Welcome. In the last session, I came up to discussing Mendel laws, and I already discussed Mendel's first law. Now, in this session, I'm going to discuss the remaining Mendel laws. And before I discuss Mendel laws, I would like to discuss about dihybrid test cross. So that was a question I asked in previous session to find out the phenotypic and genotypic ratios of dihybrid test cross. Okay, so before that, we will also see again the what is test cross with an example. Then I will even explain how the dihybrid test cross is performed and what will be the phenotypic and genotypic ratios are. So let's see the test cross. So here the test cross helps in finding out okay genotype. So that is, it's an unknown genotype. So when we do not know the genotype of an individual, then the test cross will help us to find out. So that is here whether it is homozygous individual or heterozygous individual. So to understand this, I will take one trait, that is flower color. So we know in case of pea plant, okay, the violet color is dominant. So violet color of the flower is dominant and white color is recessive. So for this, I'm going to take here capital W for violet color and small w for white color. So this is dominant and this is recessive. So here I have an individual, so its genotype is unknown. So genotype is unknown. So it is whether it is capital W or capital W, okay, small w. So which one is this, the genotype of an individual? So how for that, how to find out, okay, it. So I told you already, we need to cross this unknown the genotype with its recessive parent. So first we will take this, okay, capital W will cross with the recessive, so that will be small w, right? So here this is, okay, plant having violet colored flowers. So pea plant having violet colored flowers, and this is the pea plant with the white colored flowers. So I'm going to cross the pea plant that has violet colored flowers, okay, with the white colored flowers. So here capital, okay, I mean W, and in this case we get okay, small w. Now, okay, fertilization, of course these are gametes n and these are parents 2n. Now what I'm going to get is capital W, small w. So what I see, all will be, so in F1 generation, so all, okay, plants will have violet flowers. So we'll have violet flowers, right? And when we get all plants with the violet flowers, so this indicates that the parent is homozygous dominant. So the result is having all plants with violet flowers. So this indicate, okay, so this indicates, okay, so the unknown genotype. So that is the unknown genotype is homozygous dominant. So that will be capital W, capital W. Okay, so it indicates the unknown genotype is homozygous when we get in F1 generation all plants with the violet colored. Okay, so that is how we interpret the information. Then we will take another one with the heterozygous condition, right? So now let us see in this. Okay, we'll take heterozygous, capital W, okay, small w. Of course we do not know what actually, whether this is heterozygous or homozygous. I'm going to cross this one. So of course to outside this is, okay, violet, okay, colored flowered pea plant is crossed with the, okay, white colored flower pea plant, right? So these are parents to N. Okay, so during the formation of gametes, capital W, and this is small w, and here we'll get small w. So gametes N. So this one and this one. So I'll get capital W, small w, and this is small w and small w. So this is F1 generation. So we can see here, okay, 50% of plants will have violet colored flowers. So 50% of okay, so 50% of plants will be having violet colored flowers. So all these will be having violet colored flowers. And coming to this 50%, okay, of plants will be having white colored flowers. When I get, okay, two such categories here, so half of them are violet colored and half of them are white colored. So in earlier case I obtained all violet colored flowers only, so that indicated the unknown genotype is homozygous. Now when we obtain this, okay, violet colored flowers, it indicates that unknown genotype is heterozygous. Okay, so unknown genotype is heterozygous. Okay, so this is how the test scores will be useful to find out, okay, unknown genotypes. Now let's come to the dihybrid test cross. So let's say the dihybrid test cross. So we know in the dihybrid test cross, the genotype, okay, so whatever that unknown genotype, we need to cross with the recessive parent. So capital R, okay, small r, capital Y, small y, need to be crossed with the small r, small r, small y, small y. So, okay, so this is round yellow and this is wrinkled green. So here parents, okay, 2n, and we know here there will be formation of 4 gametes. So there will be formation of 4 gametes. So capital R, capital Y, okay, capital R, small y, then small r, capital Y, and small r, small y. Okay, so we get total 4 gametes, and here we get only one type of gamete, small r, small y. Okay, so gametes n. Okay, let's put them in the Punnett square. Okay, on one side the male and one side the female gametes. Okay, so capital R, capital Y, capital R, small y, small r, capital Y, and small y. So here small r, small y. So let's see how we get. So capital R, capital Y, small y, capital R, small y, small y. So small r, capital Y, small y, small r, small y, small y. So here you can see in this case it is round yellow, and here we get okay, round green. In this case we get okay, wrinkled yellow, and in this case we get wrinkled green. Okay, so this is the F2 generation. So this is F2 generation. Now let's look at the phenotypic ratio. The phenotypic ratio of dihybrid test cross. So the phenotypic ratio, let's see here how many we have is a round yellow. Okay, how many we have is round green, then we have is a wrinkled yellow, and another one is wrinkled green, right? So here the round yellow we have is 1, then okay, the round green is 1, the wrinkled green yellow is 1, and wrinkled green is also 1. So hence the phenotypic ratio will become 1:1:1:1. So that is the phenotypic ratio of dihybrid test cross. Now coming to the genotypic ratio. So the genotypic ratio. So here coming to the genotypic ratio, so here capital R, capital Y, small y, we have only 1. Then capital R, small small y, we have 1 here. And then okay, small r, capital Y, small y is also 1, and small r, small y will be also 1. So hence in this case also the ratio is 1:1:1:1. So the dihybrid test cross ratio, right, that is both phenotypic and genotypic ratios will be 1:1:1. So that's about okay, the dihybrid test cross. So now we'll move on to the Mendel laws. So here already in the last session I discussed the first law. Okay, so here Mendel's first and second laws. Okay, so these first and second laws, so he has given it basing on the observations of monohybrid cross. So these two laws were based on okay, so based on observations of monohybrid cross. So observing the monohybrid cross, Mendel proposed first and second laws. And coming to the third law, it was based on dihybrid cross. Based on dihybrid cross. So in the last session, I already finished about the first law. So now we will see about the Mendel's second law. So that Mendel's second law is called law of segregation. So here segregation, which means a separation. So law of segregation, okay, also called law of purity of gametes. So it's called law of purity of gametes. So Mendel's second law, or law of segregation, or law of purity of gametes. So here let's see what actually it means. Now when a cross is done involving, so when a cross is done involving a pair of contrasting characters, involving a pair of contrasting characters. So when a cross is done involving a pair of contrasting characters, what happens is both alleles stay together in F1 generation. So both the alleles stay together in F1 generation, okay, without mixing or blending. So without mixing, so I'll use the word mixing. Of course, we can use also word blending. Later I will explain what actually is that mixing or blending. So both alleles stay together in F1 generation without mixing, okay. And when F1 hybrids are self, okay, crossed, right, and when F1 generation is self crossed, so when we do this self crossing, okay, each allele separates, each allele separates, okay, from one another. So it separates from one another without change. So is self crossed, and okay, when it's self crossed, each allele separates without change. So what actually we mean by it? So we know, I'll take monohybrid cross. Okay, so when a cross is done involving a pair of contrasting characters like tall and dwarf. So when we have taken this, okay, so here the gamete formation, and we see the production of F1 generation, okay, capital T, small t. So when we have seen this F1 generation, so when we see this F1 generation, we can see that the two contrasting, okay, alleles, they are staying together in F1 generation, and now they are not, okay, affecting others expression. So this capital T is not interfering with the small t, or small t is not interfering with the capital T. So both remain together without affecting. So that's what, without mixing or blending. They don't, okay, blend or mix and produce another type of another phenotype. So that is not seen. Okay, so that is what Mendel observed. Okay, so this character, the tall character and dwarf character, were present together, okay, and they did not interfere with one another or mixed with one another, and they, okay, remained separate. So that's what, that is the reason why he also called purity of the gametes, right? So they were pure. Now when this F1 generation is self-crossed, so when they have done the self-crossing of the F1 generation, so what they have observed is that, okay, these two, okay, alleles, they got separated. Okay, so these two alleles, they separated, okay, without change. So they did not change anything. So this capital T, that is, the capital T will indicate it is tall. So it is tall only. So here you can say that if, what happens if they interfere, say if they interfere or if they mix or blend, then we can see a plant that is not too tall or dwarf. So means we get an intermediate form, but we did not get, I mean, when Mendel was doing, he did not get any intermediate, okay, height. So either he got a tall or he got, okay, dwarf, but he did not get any intermediate form, okay, or intermediate height. So that's what capital T, of course, you can see they got separated, okay, and finally you can see here, this is the dwarf plant and these are all the tall plants. So the capital T and small t alleles remained, okay, separate without mixing with one another. So that is what the Mendel's second law, or law of segregation. So here segregation means, okay, separation. So that's about the Mendel's second law. Now let's look at the third law, the law of independent assortment. The next one is law of independent assortment. So let's see, first is definition. So here, when two pairs of traits are combined in a hybrid, when two pairs of traits are combined in a hybrid. Okay, so of course that's what I'm talking about, capital R, capital Y, small y. So when two pairs of traits are combined in a hybrid, then segregation, that is separation. So segregation of one pair of characters, is independent of another pair, is independent of another pair. So when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of other pair of characters. So you can see here capital R, capital Y, small y. Okay. So here the separation of this pair is independent of this pair. So right, I'll take here capital R, capital Y, small y. So we know that during the formation of gametes, okay, this capital R, capital Y, and capital R, small y, then small r, okay, capital Y, and this is small r, small y. So we can see there is a formation of four gametes. So each one actually contributing to 50%. So here the capital R. Okay, so here the segregation of 50% of this capital R and 50% of a small r. So this and this have 50%. Okay, and this capital Y and small y will also have 50-50, okay, of separation. So it is independent. So this is independent of this, okay, small r. Capital R, okay, it has a 50% of chance, and this small r also has 50% of chance. Okay, so that's what when we take here, you can see here this is of 50%, especially I'm talking here in case of capital R, okay, and the small r. And you can also see here the capital Y and also capital Y, which is all okay, are also 50-50, okay, percent of separation. And thus okay, so it results in four types of pollen or four types of eggs. But okay, in that case, okay, so we need to take each one with the 25%. So this is okay 25%, so this is 25%, and this is 25, and this is okay 25%, right? So here, okay, separation of these two and separation of these two is 50-50, right? That's what here, when two pairs of traits are combined in a hybrid, so this is a hybrid, the segregation of one pair of characters is independent of okay another pair of characters, right? So of course these are all gametes, right? And here we can see that okay the frequency of each of the gamete is only 25%, is of okay only 25%. So right, fine. So that's all about the law of independent assortment. Now, so that's all about the Mendel's okay experiments and laws. Now after Mendel, many other scientists followed the same way how Mendel did experiments. So by taking the homozygous conditions, then crossing them, getting F1 generation, and this F1 generation will be again self-crossed and look in the F2 generations. Now, okay, the results obtained by the other scientists, okay, doing in the same way as Mendel, okay, they did not get like Mendel's results, so they were different from the Mendel. So that's what we call them as deviations from Mendel laws. Okay, so deviations from Mendel laws. So let's see the deviations from Mendel laws. So under this, first I'm going to discuss about incomplete dominance. I'm going to discuss about incomplete dominance. And this incomplete dominance was discovered by Carl Correns. Was discovered by Carl Correns, okay, when he was doing experiment on 4 o'clock line, okay, on, I mean, discovered by Carl Correns in, okay, 4 o'clock line. So whose scientific name is Mirabilis jalapa. So its a scientific name is Mirabilis jalapa. Okay, so he found this incomplete dominance in 4 o'clock plant Mirabilis jalapa, and this phenomenon is also found in, okay, also found in another plant called snapdragon, okay, also called dog flower plant. So snapdragon or dog flower, so whose scientific name is Antirrhinum majus. So its scientific name is Antirrhinum majus. So in that plant also we find this phenomenon called incomplete dominance. Okay, so here what Carl Correns did is he has taken, okay, the flower color, the character, okay, flower color, and what he observed, it has two alleles. So one he found with red colored flowers, and okay, another with white colored flowers. Okay, so the flower color in these plants, either in Mirabilis jalapa or in Antirrhinum, he found that, okay, the red colored flower plants and white colored flower plants, and he observed that the red color is dominant. So hence we'll take capital R. So this is dominant, and the white colored flowers are recessive. So hence I'm going to take a small r. So this is recessive. So red colored flowers, so they are dominant, and white colored flowers, they are recessive. Okay, now what he did, okay, he has taken homozygous dominant red colored flower plants and crossed with homozygous white colored flowers. Okay, so let's see it. So he crossed, okay, caplar, so this is homozygous red and crossed with the homozygous, okay, white, and these are parents, they are diploid. And during the formation of gametes, you can see here this is capital R, and okay, this is small r, so we get only half the number of genes into the gametes. Okay, so these are gametes which are haploid. Okay, so meiosis and meiosis, and then, okay, fertilization or syngamy, okay, results in the formation of capital R, small r. So this will be the F1 generation. Okay, so that will be again deployed. And what he observed is that he did not get red colored flowers. So actually the okay R is dominant, and I mean capital R is dominant and small r recessive. So when the dominant character is present, okay, it should actually express the red, but it was not red, but it was pink colored flowers were observed. So it means neither white nor red were seen in case of F1 generation. Okay, so if it was according to Mendel, actually one of the allele or a factor will get expressed and the other one will not get expressed. So in this case it was not at all following the Mendel laws. So that's what I have mentioned. Okay, deviations from Mendelian laws. Now, okay, Karl Correns followed, okay, like Mendel. So after doing this, he gone for self-crossing the F1 generation. So let's see the selfing of F1 generation. Okay, so self-crossing F1 generation. So what they have, that is taken, so capital R, small r. So this is pink colored. So plants having pink colored, okay, flowers. And again plants having pink colored flowers. So these are parents, okay, 2n. Now we know it results in formation of two types of gametes. So capital R, small r, and here also capital R, small r. So these are gametes n. Okay, so now we will put it in Punnett square, so it will be easy to understand. So I'm going to take the Punnett square. So capital R, small r. So in this case, capital R, small r. Okay. So this and this we obtain, okay, red. So this and this we obtain pink. So this and this capital R, small r, again we obtain pink, small r, we obtain, okay, white. So right, so this is the result in the F2 generation. So F2 generation. So now let's look at the phenotypic ratio of incomplete dominance. So phenotypic ratio of incomplete dominance. So here we need to know how many are, okay, red, how many are pink, and how many are white. So we can see here there is one red, two pink, and one white. So hence the phenotypic ratio of incomplete dominance will be 1:2:1. So it is the phenotypic ratio of incomplete dominance is 1:2:1. And coming to the genotypic ratio. So genotypic ratio of incomplete dominance. So let's see in this case how many are capital R, how many are capital R, small r, and how many are small r, small r. So here you can see there is only one capital R, small r, there are two, and small r, one. So hence the genotypic ratio will be also 1:2:1. So in this case, okay, both the ratios are 1:2:1. But in case of Mendel's monohybrid cross, which involves same, so this is also a monohybrid cross. So here only one character is differing. Okay, this is also monohybrid cross, but Karl Correns did not get the ratios like Mendel. Okay, So there, in the tall and dwarf characters, Mendel what observed is the phenotypic ratio was 3:1, but we obtained the phenotypic ratio here as 1:2:1. Right. So now the question comes that what actually is dominant, how the genes become dominant, and how they act as recessive. So let's see the concept of dominance. So what actually is dominance or recessive, or why some alleles are dominant and why some alleles are recessive? Okay, so let's see the concept of dominance. Okay, so here before we discuss, in a diploid organism, so in a diploid organism, so in a diploid organism, there will be two copies of each gene. There will be, or there are two copies of each gene. So in a diploid organism, there are two copies of each gene. Okay, so those two copies, okay, of each gene is what we call, okay, a pair of alleles. So that is pair of alleles. Okay, so that's what when I have taken the parents, I always mention for each character, okay, two alleles. So capital T, small t, or you can see here capital R, capital R, capital Y, capital Y. So here, right, as I said, these are two copies of each gene. So this will be the pair of alleles, and this is another, okay, pair of alleles. For every gene we find, okay, a pair of alleles. Okay, so let's see how some alleles will act as dominant and why some will be acting as right, recessive. We'll take an example to understand this, okay, dominance, the concept of dominance. So here, say suppose there is an enzyme, okay, and that enzyme will be coded by, okay, gene, and this gene has two alternative forms. Say this is allele 1 and this is, okay, allele 2. Okay, so these two are alternative forms of a gene. So that's what I mentioned. There are two copies of each gene. Okay, so these are two copies of each gene, right? And right now, says this one allele, this is the normal, okay, gene, and this is a modified form of the normal one. So this is a normal one, and the normal allele will produce enzyme. So it produces, okay, functional enzyme. It produces functional enzyme. Now coming to this modified form, so it has okay, different possibilities. So what are different possibilities is that this modified, okay, gene may produce normal or may produce functional enzyme. So it may produce functional enzyme. Okay, so that is one possibility. Or it may produce less efficient enzyme, may produce less efficient enzyme. So that is another possibility. And another possibility, it may produce may produce non-functional enzyme. It may produce non-functional enzyme. And okay, and another possibility is that it may not produce any enzyme. It may not produce any enzyme. So these are four possibilities with the modified gene, whereas the normal gene, okay, will be producing all the time the functional enzyme. So that functional enzyme is going to convert the substrate S to, okay, products. So with the help of the functional enzyme, right? So here this modified form is a copy of this normal gene. Okay, so I'm mentioning that there are two copies of each gene and they will be present, okay, in the form of alleles. So this is these two are alternative forms of a gene. Okay, and say suppose this one is producing, allele 1 is producing functional enzyme, and this is the normal or the original gene that is present. So I'll mention as original allele, and from this in course of time have resulted in the formation of modified, okay, allele, and this one may produce the functional, okay, enzyme as it is. So at present it's quite difficult to understand that why these many possibilities are present. So that we will understand only when we study the next chapter, the molecular basis. We need to know the molecular basis, okay, to understand why it has many possibilities. So after understanding the molecular basis of inheritance, then this concept will be clear. So at present you need to remember that the modified allele will have, okay, right, different possibilities, like produce functional enzyme, so may produce less efficient enzyme, may produce non-functional enzyme, or may not produce enzyme at all. Okay, so this normal one or the original allele, this will act as dominant allele, and the one which is modified, so this will be acting as recessive. So the original normal gene is the dominant one, and the modified one will be the recessive one, right? So with that I give a statement that, okay, the phenotype or trait, the phenotype or trait, so this will be dependent on the functioning of, so it depends on functioning of unmodified allele. So the phenotype or trait depends on functioning of unmodified allele. Okay, so this phenotype or trait depends on functioning of unmodified allele. Okay, right, and the unmodified allele, which actually is the original allele I mentioned, okay, that will be the dominant allele, and the one which is modified, so this will be, okay, the recessive allele, will be the recessive allele. Okay, so this is important. The phenotype or trait will depend only on functioning of unmodified allele. Right, so hope it's clear. Right, so again I'll repeat. So allele 1, that is original one, okay, will be acting as dominant, and the modified one is generally recessive, so it will become recessive allele, and that recessive allele have, okay, several possibilities. Okay, so to understand this, we need to know at molecular level that we will understand in the next chapter. Okay, so at present, keep in mind that the normal, okay, unmodified allele will be the dominant, and the okay modified one will generally become recessive. Right, so with this, I will end today's session. So we'll see in the next session co-dominance.